Now, the skin is an organ of tactility. It allows us to feel our environment and to sense our environment in different ways. And this is based on peripheral sensory receptors. So in the dermis there are various nerve fibres and various nerve corpuscles, and what these all do is they detect some environmental external stimulus on the surface of the skin, and when they detect the particular sensation that they are sensitive to, what they will actually do is generate a brand new nerve impulse. And that new nerve impulse will then go off to the cord if it's the lower part of the body, but either way it'll go to the brain. If it's the face or the top part of the body, it'll go via a cranial nerve. So the skin contains peripheral sensory receptors. All of these peripheral sensory receptors are sensitive to particular types of stimulus, and they will all work by generating a new nerve impulse, sending that information to the brain. And all that information will go to the sensory cortex of the brain. So if it's the right side of the body, that information will end up in the left sensory cortex, in the parietal lobe of the brain. If it's the left side of the body, that information will end up in the parietal cortex on the right side of the brain. So it's actually felt in the brain, but the nerve impulses are generated in the dermis. So let's look at how this works to help our understanding of the sense of tactility. So here we have the surface of the epidermis as usual, and going down into the dermis we have a hair follicle. And here we have the junction between the epidermis and the dermis that we're familiar with. And here's the hypodermis here. Some of the hair follicles go right down into the hypodermis actually. So epidermis, dermis, hypodermis. Now we already know, because if you've watched the first part of this DVD, in the very bottom layer of the epidermis there are small sensory cells called Merkel cells, and these Merkel cells are connected to Merkel cell discs, and these join together and carry the information off towards the brain, and these are sensitive to light touch. So the Merkel discs, the Merkel cells, are detecting information via the epidermis. They're the only nerve fibres in the epidermis. The rest are in the dermis. And to look at it very basically, there's two types of peripheral sensory receptor in the dermis. There's those with corpuscles, where there is some specialised structure detecting the tactile stimulus and generating the new nerve impulse. And there are those which are just free nerve endings penetrating the dermis. So let's think about the corpuscular peripheral sensory receptors first. And these are called mechanoreceptors. They are detecting mechanical stimuli. They are mechanoreceptors. And the first are up in the dermal papillae, up here, and they're called Missner corpuscles. Missner was a German anatomist who first discovered the Missner corpuscles. And these are moderately small corpuscles, but they're found in the dermal papillae, near the surface of the dermis. And the reason that they're near the surface is these mechanoreceptors detect touch, often fairly light touch. Maybe not as light touch as the Merkel cells, but the Missner cells detect touch. So when someone's just touching you, that's the Missner cells that are detecting that. So they're fairly near the surface. And again, these nerve fibres will come together and they will go off towards the central nervous system. This fibre here will actually be a sensory neuron. It will actually be a dendrite of a sensory neuron. Because remember that a dendrite is any nerve fibre carrying information towards the cell body. So the cell body for these, if these are in the body, the cell body will be near the spinal cord. So these are the dendrites taking this information. In this case, because these are Meissner corpuscles, about touch. So we know the skin is sensitive to touch, and the skin is also sensitive to pressure. So deeper down, very often actually in the hypodermis or deep down in the dermis, there are really quite large receptors. And they look a bit like an onion if you slice them. They've got layers. Because they're actually made up of specialised Schwann cells, much as composes the myelin sheath in the peripheral nervous system. So these are large laminated sensory corpuscles, and they're called Pacinian corpuscles. Pacinian corpuscles. And they're deep down. And I think you can probably see they're laminated. They're deep down. And again, these are connected to the dendrites of sensory neurons, which take mechanical information off towards the brain. And the Pacinian receptors detect pressure. It's not touch, it's pressure, when there's a pressure on the dermis. And again, it makes sense that these are near the base of the dermis, because you only want them to fire off to generate new nerve impulses when there's a significant pressure. So they're corpuscular receptors. So we've got touch, we've got pressure. And the skin is also sensitive to temperature. So there must be thermoreceptors. And of course there are thermoreceptors. And in fact there's two types of thermoreceptors. Some specialised thermoreceptors detect it when the skin gets too cold. Other specialised peripheral thermoreceptors detect it when the skin gets too hot. So there's cold thermoreceptors and there's hot thermoreceptors. And these are just free nerve endings in the dermis. They're not corpuscular like these ones were. So they're just free nerve endings. So this could be one here for branched free nerve endings for heat. Again, going off towards the central nervous system. And this could be one for cold, just next to it. Again, going off towards the central nervous system. So the thermoreceptors detect heat and they detect cold. They're specialised thermoreceptors. So we've got touch, pressure, temperature, and of course you know that the skin is also sensitive to pain. There are specialised pain receptors. And the specialised pain receptors are called nociceptors. Nociceptors detect pain. They are detecting noxious stimuli. They are nociceptors. And again, these are just free nerve endings in the dermis. So here we have nociceptors. It is the presence of the nociceptors that is making the dermis sensitive to pain. And of course the nociceptors don't go into the epidermis. It's the dermis which is sensitive to pain. This is why partial thickness injuries are so painful. So for example, if you have a partial thickness burn where this top part of the epidermis and top part of the dermis is burnt, you're exposing the nociceptors. Or if you have a graze where there's a partial loss of the tissue, again you're exposing the nociceptors, and that is very sensitive because the nociceptors are detecting pain. Free nerve endings penetrating through the dermis. They don't like being cut, they don't like being torn. Anything that starts to injure the tissues will generate a new nerve impulse in the nociceptors. And the threshold of generating a new nerve impulse, what we call the depolarization threshold, for nociceptors is actually precisely set at the point where the tissues start to be damaged. It's remarkably clever. So if you warm the tissue up to 45 degrees centigrade, the nociceptors will start to generate a new nerve impulse, because that's the point at which the temperature can start to damage the tissues. Or if you're pressing on the skin hard enough to start damaging the tissues, at the point where the tissue damage is just starting, the nociceptors will generate new nerve impulses. And we mentioned the term depolarization there. What actually happens is there is a reversal of the polarity in these nerve fibers, and that's what the new nerve impulse is. The new nerve impulse is generated when these depolarize and travels off to the brain. In this case, they go up the spinal cord to part of the brain called the thalamus, where the pain is generated, and then that pain is sent to the sensory cortex, so we know what part of the body the pain is coming from. But again, for the purposes of this talk, the nociceptors, the pain receptors, are free nerve endings branching throughout the dermis, making it sensitive to pain. So we've got touch, light touch, slightly heavier touch, pressure, thermoreceptors, pain. Now there's a couple other types of receptors I think I will mention. One sort doesn't work very well in me anymore, but it certainly did when I was a child, and they're tickle receptors. And again, the tickle receptors appear just to be free branched nerve endings. I guess adults still can be tickled in the right circumstance. Free branched nerve endings in the dermis generating a sensation of tickling. And also, there's another type of branched free nerve endings, and these are sensitive to itch. So when you're itchy, these are generating that sensation of itch. And if you work in clinical practice, you will come across patients for whom itching is a real problem. For example, in allergic reactions, inflammatory mediators will be released into the dermis, and that greatly stimulates the itch receptors. So an acute itch, we think, oh, I wonder if that could be an allergic reaction, because it could be. And we need to think about that and treat it accordingly. And the other patients that I've looked after who have the most appalling itch, terrible, distressing itching, is patients with jaundice. When patients go a yellow colour, you get bile salts accumulating in the dermis. That's why the patients go yellow, because of the bile salts. And that makes these patients very, very itchy. So anything you can do about that is a good idea. So we've got these different peripheral sensory receptors. We've got touch, we've got pressure, we've got temperature, we've got pain, we've got tickle, we've got itch. But all of them have this in common. When the appropriate tactile stimulus arrives on the surface of the body, these specialized detectors detect that. They generate a new nerve impulse, a brand new nerve impulse, that goes to the brain, and the brain cleverly detects it as that appropriate somatic tactile sensation.